Let's start out. So the central nervous system starts to form at around 2 weeks old. The birth of the average brain is about 350 grams. Brain waves have been recorded at around 40 days. So sometimes when we talk about abortion, people want to know when the brain waves are, and so that's about 5 weeks after conception. And what's kind of sad about that is a lot of people don't know that they're pregnant at that point, and if they're partying a lot and so on and so forth, we're going to find out that drugs can really affect the brain, and a lot of the development will happen just in the very beginning of pregnancy, sometimes before people even know. But here's something really interesting. At the end of the first year, the brain weighs 1,000 grams. So a lot of people tend to think that the first year of life, the baby's not really capable of much, and they kind of just leave them sitting in the crib and so on and so forth. But look at this. This is your biggest area of brain growth, 350 grams to 1,000 grams in the first year of life. So all that touch, contact, talking, so on and so forth, that's really stimulating a lot of the different dendritic pathways. The adult brain is only 1,200 grams. So basically from this slide, the main thing that I'd like you to get out of it is that the central nervous system begins to form at around 2 weeks old. That's like right when a woman would be knowing that she missed her period. And then it starts at 350 grams, but you have the biggest time of growth during the first year of life, and then the adult brain is only 1,200 grams. Okay, let's go to the next one. This is the formulation of the brain here as it starts out, and then it kind of curls into a tube, and then this becomes the forebrain, the midbrain. I'm going to see if I can find a video to show this process. Okay, this is just a picture to show you of a little embryo. So right after the first 2 weeks, basically the baby's just a ball of cells, and it becomes this blastocyst, and then the blastocyst will roll down and it will attach to the uterus, and for the first 8 weeks it's called an embryo, and then at 9 weeks it's called a fetus. But basically all the major organ systems are developing in the first 8 weeks of pregnancy. Okay, so unfortunately, this is a slide where you just kind of have to copy it down and then kind of memorize it. But this is the process in the development of neurons. So first what they do is they proliferate, and we're going to be going over stem cells later on, but they start as a stem cell. And then if you remember the radial glia that we learned about in chapter 2, they actually will direct the neurons where to go to during migration. So first we have to produce them, proliferation. Then they have to move towards their eventual destination. So as you can see, they have to go to the different places, the forebrain, the midbrain. Then they differentiate, and so first they're made, then they move, then they differentiate. So I always picture like little hands sticking out, like the little axon starts to pop out of the cell and the little dendrites pop out of the cell. And then what they're going to do is throughout life so these three right here, proliferate, migrate, differentiate. This is usually going to happen early in life, but then throughout life what you have is you have myelination. So that's like the glial cells, they produce the myelin sheath, and the more myelin that's around a certain pathway, the thicker it becomes, and that's the stronger pathway, the thicker pathway, the faster pathway. And then also what you're doing is you're constantly making new synapses throughout life. So these two, myelination and synaptogenesis, they're happening throughout life. This is kind of what learning is. This is what experience is the myelination and the synaptogenesis. So this might be a place in the video where you have to pause. You might want to click pause, because if you didn't get to write all this down, you might want to. So first they have to be built, then they have to move, then they have to differentiate, then myelination and synaptogenesis happen throughout life. But I would like you to know this process. So this might be a point right here where you have to stop the video and copy this down if you haven't had enough time. Okay, So basically, I spent some time before doing this video looking for a ball of clay. I wanted to have a ball of clay, and then I wanted to have a sculptor, like have a fast forward video of a sculptor sculpting out like a face out of the ball of clay. And he would be pruning it away, or she would be pruning it away slowly, slowly, slowly. But I couldn't find anything that I really liked. Too bad, because that's what I wanted to show you, is that basically you're born with more neurons than you're ever going to use. And then whatever you don't use, they just die. And so throughout life, there's different time periods where you have cell death. So it's either use it or lose it. So you have lots and lots of cells, and then the ones that don't seem to be used are just going to die. So I think some of you know, like some of you who are Hispanic, you know, you kind of like my Hispanic friends, they always make fun of me because even though I can speak a little bit of Spanish and I can understand a lot of what they're saying, is whenever I try to make the double R sound, the R, they always laugh because I can never do it. And then there's a sound in Asian, actually, there's a different sound that Americans can't say because if you're not exposed to those sounds at a certain time in life, then you just lose the ability to even do them. And anybody will tell you, like, if you learn a language later in life, you can always kind of tell whether or not they're a native speaker or whether or not they learned it because they just don't have quite the right accents. So we start out with more neurons, then we ever have like the picture of a big, big ball of brain, and then whatever dies, you know, whatever isn't used, then it just gets pruned away. Okay, so what determines whether or not a neuron is going to survive or not? So each cell comes with a suicide program named apoptosis, a program mechanism of cell death. And so this thing called nerve growth factor, it inhibits apoptosis. Neurotrophins are a form of nerve growth factor. They're chemicals that promote the survival and activity of neurons. So in order for a neuron not to die, it has to receive a form of neurotropin. It could be the nerve growth factor. So if that receives it because it's been used actively, then it's not going to commit apoptosis. But if it does not receive that factor, then basically what it's going to do is it says, okay, I'm not being used, so I better just die. So we're going to see that this is really important because when you're raising your children, that's the reason why I chose to do this chapter. Is some teachers don't do this chapter. Like in my school, I think I'm the only one that does this chapter, you know, because there's so many chapters that we have to go over that a lot of times they'll skip certain ones. As you can see, I chose to skip a few myself. And so this is one that sometimes they don't do, but I think it's such an important chapter to really understand what's going on in your children's brains when you have children.
This animation shows brain development from conception through birth. In the first 4 weeks after conception, neural tissue known as the neural plate forms in the outermost layer of embryonic cells. The neural plate folds to form the neural groove, then curls to form the neural tube. This tube differentiates into the forebrain, the midbrain, the hindbrain, and the spinal cord. The forebrain will develop into the cerebral cortex, which translates sensory stimulation. It also controls complex behaviors, thoughts, memories, and problem solving. The midbrain will develop into a neural relay station for sending information from the body to various sites in the brain. The hindbrain will control the most basic physiological processes, such as breathing and heart rate. The spinal cord is the pathway for conveying information between the brain and the rest of the body. Between weeks 4 and 8, the embryo grows rapidly and the face becomes recognizably human. The eyes on the side of the head begin to migrate forward and 2 distinct hemispheres of the cerebral cortex emerge. Between weeks 8 and 26 of the fetal stage, the cerebral cortex grows to cover the midbrain. By week 28, a major structural change begins. The cortex expands greatly in surface area and becomes wrinkled and folded inside of the skull. Beginning at week 28 and continuing through week 40, the brain surface fills with hills and valleys, called gyri and sulci. These cerebral folds and wrinkles help to create a vast neural network which allows the brain to become a highly flexible yet precise control center and processor of information. By week 8, the structures of the brain are already in place, but the amount of brain tissue is rapidly increasing as many new nerve cells are formed. The formation of new cells is known as neurogenesis or neural migration. During neurogenesis, new nerve cells migrate from the innermost layers of tissue outward. Like gymnasts climbing ropes, the young cells are guided to their final destination by fibers from another type of cell, the glial cell. Glial cells form the underlying structure of the nervous system. Although earlier formed cells rest closer to where they originated, later formed cells move further away from their starting point. In this way, the brain grows, adding layers to itself like the skins of an onion. Once each nerve cell reaches its destination, it begins to acquire characteristics specific to that area of the brain. At this point, each nerve cell sprouts the extensions it needs to communicate with other neurons. There are two types of extensions, dendrites and axons. Dendrites look like a network of antennas and receive incoming signals. An axon is a single, larger extension which carries outgoing signals to other neurons. Synaptogenesis is the process in which synapses proliferate in the brain. A synapse is a tiny gap between the dendrites of one neuron and the axon terminal of the next. It's the point of communication between two neurons. So many new axons and dendrites form during the process that it's sometimes called exuberant synaptogenesis. This cell growth process continues into a newborn's first year of life. For an information signal, or impulse, to move from the axon of one neuron to the dendrites of another, it must cross the synaptic gap. In this animation, an impulse looks like a green ball of light. When an impulse reaches the end of an axon, a chemical called a neurotransmitter is released. In this animation, neurotransmitters look like red balls of light. Neurotransmitters allow an impulse to cross from the axon of one neuron across the synapse and onto a receiving dendrite on another neuron. To create a more efficient and rapidly working brain, some of the newly formed synapses are eventually removed. Synaptic pruning occurs during the fetal period, infancy, childhood, and adolescence. It's believed that pruning is a way in which the brain becomes customized to an individual's nervous system as a response to that individual's unique experiences.